Uh, so I think the recording has just started, so maybe now is a good time for us to start. It's a pleasure to welcome back for the second of his three lectures, Amaury Lambert. He's going to be speaking to us today about the formation of new species, and I'll send around in a little while, for those who joined a little late, the link to his talk in case you want to download and follow along. And we just, I think, have posted the link for the first lecture talk video and the slides. So anyway, let me transfer this over to Amaury, and I'll enjoy his lecture.
Thank you very much, Ivan. So yeah, as you can remember, the title of the series is about evolutionary processes and patterns of biodiversity. And last week, we saw some models for the expected diversity, genetic diversity that you could observe in a sample of DNA sequences from the same species at the same locus and maybe at different loci, and how recombination did couple these genealogies at different loci, and also how the information on the diversity in this sample could inform us about the past demography of this species. And today I will deal with what happens when you take, instead of the same, to take individuals from the same species, you take individuals from different species and try to characterize the diversity that you observe. So let me start with a few concepts. So in systematics, which is the science of, let's say, sorting individuals into species, genera, families, classes, orders, in such a way that is consistent with their evolutionary history, species in their biological definition are groups of individuals which can interbreed. It's also a synonym for mate or hybridize, and cannot interbreed outside the group. The term that is used is reproductive isolation. And so the basic question that I want to address today, and that I've been interested in from, let's say, modelers' point of view, is how do new groups emerge from old groups? Of course, this is relevant to this series of lectures because reproductive isolation is a very powerful engine for fueling diversity. It prevents hybridization from blending phenotypes. So instead of having a gradient of all possible body sizes, you might have a species with very different body sizes. And of course, when you sample a pool of individuals from very different species, from many different species, you will have a lot more diversity than when you sample them from a single species. Here, so let's say that last week we talked about the diversity at a sample of DNA sequences and how this diversity was mediated by the genealogy of the individuals that you had sampled. And today I will talk directly about the genealogy of these individuals, which is called the phylogeny, for the genealogy of species. And you'll see that we consider phylogeny from diverse aspects. The tree shape, so the combinatorial properties of the phylogenetic tree, and the edge length, so the metric aspects of the tree. On the right panel, you can see an example of a big tree. It's called the tree of life because it's supposed to encompass all forms of life. Actually, only a few representative from a few species, and from that tree, you can see in blue the importance of bacteria and archaea in the living kingdom. And a specific question that I will address is whether we can learn from the phylogeny about the diversification process. That is the process that is responsible for the emergence of new species and the extinction of old species. So these two aspects that I mentioned, the combinatorial aspect and the metric aspect, are embodied in two very popular measures used by macroevolutionists. One is beta and the other is gamma. As you can see from the cartoon, beta is negative for trees that are imbalanced, and beta is positive for trees that are balanced. Okay, so I will talk in the last slides of the talk. I will be a little more specific about beta, but from now on, just remember that beta is supposed to be 0 trees like the Yew tree, the pure birth tree, and the Kingman coalescent that I introduced last week. Gamma is a measure of relative branch length. It's positive for trees which has nodes close to the tips, and it's negative for trees which have nodes close to the root. And so Kingman coalescent has nodes close closer to the root, so it has closer to the tip, so it has a positive gamma. The Yew tree has a 0 gamma, and as you can see in red, real trees are known to have a negative beta, so more imbalanced than what is predicted from neutral models, and negative gamma, which is different from the Kingman coalescent and more and close to. I'm sorry, I have a problem with my. So that's for beta and gamma, and I won't linger much about these measures, but I can't resist the pleasure of showing you some of these phylogenetic trees or phylogenies. Here on the right, you can see the phylogeny of gymnosperms, which is a branch of seed plants encompassing mainly conifers. And you can see on the top of the tree, you can see a very small subclade, subtree, which is deeply rooted and only counts one species, the Ginkgo biloba. And this feature and this phenomenon of having this small, deeply rooted subclade at all scales actually is responsible for the imbalance of empirical trees. There are many examples of such small and deeply rooted clades. Here, for when you zoom in on the gymnosperm phylogeny for conifers, you can see also in yellow this small clade. In the phylogeny of birds, you can also see that Paleognathai on the right of the circle. Here you see that the tips of the phylogeny are arranged on a circle, and the root of the phylogeny is in the center. These relatives of ostriches and emus and kiwis is a very small and deeply rooted subclade also in the bird phylogeny. And maybe the most conspicuous example of such a very small subclade deeply rooted in the phylogeny is the case of monotremes, which has only the four species of echidnae and the single species of platypus. And you can see in black on the right of the phylogeny of mammals. So let's go for the outline. So in the first section, I will talk about models of diversification which are lineage-based, where the species are the elementary particles of the process. And I will introduce a dual object to the birth-death process called the coalescent point process, analog to the Kingman coalescent. I will then show you the results of 3 works inferring diversification from phylogenies. And then I will come to more microscopic models of speciation, where now we want to account for the progressive emergence of reproductive isolation and open the species box. Along the way, I will talk about the interbreeding graph, the graph whose edges embody the ability to interbreed between populations. And then I will be a little more specific about this puzzle of the imbalance of empirical phylogenies. So in what is called the lineage-based model of diversification, species are seen as the elementary particles of the system that can independently split at speciation events and die at extinction events. So it's basically a branching process where the birth and death rates, B and D, may depend upon 4 classes of variables, T, N, A, and I, where T stands for time, absolute time, N stands for the number of co-occurring species in the system. A is a non-heritable trait, a trait whose value is drawn from a given distribution at each birth event, like age, which is, as you know, reset to 0 at each birth, or a heritable trait. Typically, a trait is a phenotype, a physical feature of a species, like body mass. And as you can see on the left panel, the trees that we consider will be oriented, meaning that the daughter sprouts to the right of the mother, and time flows upwards. The most celebrated model of such a birth-death process is the Yule model, also called the pure birth process, where B is constant and D is zero, no extinction. As you can see from this, in this model, there's no information on the process of speciation, but a very nice feature is that it plainly generates a phylogeny from the knowledge of births and deaths occurring through time. Now, of course, we don't have the whole information about the phylogeny, about this birth-death tree, but the only information that we have is about the phylogeny of extant species. Extant means alive, okay? And so if we want to be more specific, we want to ask, what is the law of the reconstructed tree under the birth-death model that I just introduced? So the reconstructed tree, or reduced tree in math, is the tree spanned by species extant at some fixed time, capital T, or possibly by a sample of these extant species. So here's what I mean by a sample. So you take the tree spanned by all species extant at T, and actually what you do is that you keep only a fraction of these by sampling independently with the same probability each species extant at time T, and you look on the right at the tree spanned by sampled species that is generated by this sample. Another thing that we can do is assume that instead of killing or removing species alive at time capital T, we will remove lineages that were alive at time capital T minus S to model the presence of a mass extinction event. We would say a bottleneck in population genetics, again by assuming that every lineage that was present at that time was removed and killed with its entire descendants with the same probability independently.
So are we assuming that we kind of know a priori when these bottlenecks occur?
Exactly.
Okay.
Exactly. I mean, it might be an unknown that you want to infer, but it's fixed. And you may have several of these. Okay? So the main result that we got in this framework is the following. So assume that you have these birth and death rates that may depend on T, N, A, and I. Recall that T is time, N number of co-occurring species, and A is an inheritable trait. If you're only interested in the tree shape, so the tree seen as a combinatorial object, then the reconstructed tree always has the same topology, the same shape in distribution as the pure birth Yule tree, as soon as you assume that B at most depends on T and N, and D depends on T, N, and A. Okay, so it means that all these trees have the same beta equals 0. Okay, they have the same degree of balance, and more than that, they have exactly the same shape in distribution as if you had a constant birth rate and no deaths. All right.
Amaury, can I ask? So I understand the T. So N, A, and I, you introduced them, but how can you have, in this model, I don't see how you have N bigger than 1, right? Because you just have these splits. So each time you split, you end up with a different species, right?
So N is the number of particles in your birth-death process, if you want.
Oh, it's the total number. Okay. And A and I, how are these determined?
So A, think of A as the age of your species. So you have one particle which has age A at time T.
So that was the time it split off from.
Exactly.
Okay. And I was what?
And I might be some traits, so some, let's say, real number that varies stochastically through time. I won't talk much more about it.
But that's some sort of possible space-time randomness, or space or time randomness, I guess.
Yeah, exactly. Yeah.
Okay.
And so, and now if you're interested in tree shape plus edge length, if you're interested in the law of the whole reconstructed tree, then as soon as B at most depends on time and D depends at most on time and age, or non-heritable trait, the likelihood, the probability of the tree, always has a simple explicit product form, which means that the tree is what is called a coalescent point process. So what is a coalescent point process? Assume you're given the law of some random variable, capital H, which is positive. Then the coalescent point process can be generated are defined as the oriented tree whose node depths H1, H2, H3 form a sequence of independent copies of H killed at its first value larger than capital T. Okay, so the way you construct, you build this tree, once you have this sequence of H1, H2, etc., a sequence with a geometric number of such independent node depths, you see that you can construct the tree by drawing each of these edges, starting from the capital T level, and then going down up to down to the length of the edge, and then drawing a horizontal line from right to left until you encounter a bold line, right? And so this, you see that this is a very simple way of building a tree from a bunch of independent random variables. And so from this, you get a super fast way of simulating the reconstructed tree. And of course, since the likelihood has explicit product form, if you have a parameterization of H, if you know how this variable H depends on the initial ingredients of your model, then you get a very simple and efficient inference method for knowing how or discovering the birth and death rates, how they depending on T and A.
So how is the distribution of H chosen, or that's what we're trying to infer?
Yeah. So yeah, sorry if it was not clear. So if you're given some H, you can construct a CPP, corresponding point process, this way. Mm-hmm. What I'm saying is that if you started from a birth-death process with B depending on T and D depending on T and A, then the reduced tree at time capital T has the same law as a CPP with some node depth distribution H. And of course, the game then is to guess the law of H from the birth and death rates, the initial ingredients of the model.
And the way that you go from the picture that you're giving to a tree is how exactly? Yeah, this coalescing point process.
So here the tree, the tree is obtained by assuming that the dashed lines have length 0, but they're connecting 2 points, two bold points together. Okay, so the dashed lines are, so you see the metric of the tree is given, it's basically a graph. You take the graph distance on this, not the graph distance, sorry, you take the usual metric on this object, and you connect points that are linked by a dashed line, but the dashed line has a length 0. Is that clear?
Okay.
Yeah, like here, right? Like here. Okay. So then the theorem is a little bit more specific than that, more precise. It tells you how the law of H can be deduced. So the law of the node depths of these trees can be deduced from the ingredients of the model. So it's convenient to define capital F as 1 over the tail distribution of H, and then capital F satisfies some linear integral differential equation, which involves B, the birth rate, and G, where G of T and S is the density at time S of the extinction time of a species born at time T. And you can guess that I didn't show you the detail here, but that it can be expressed in terms of B and D quite easily. And actually, the result still holds if you assume that there were missing species, that you have not sampled all species, and there were mass extinction events along the way. On this slide, I listed a bunch of special cases. Maybe you can come back to that slide or to the initial paper after recording. So these special cases include when B and D only depend on time, when B is constant and D only depend on age. So actually saying that D depend on age depends on, or saying that D does not depend on age means that the lifetime of a species is exponentially distributed. Now, saying that D depends on age is saying that the lifetime of a species can be general, okay? And so in this case, actually, you can use another trick, which is the contour process of the entire tree, and show that this contour process is actually what is known as a Levy process. And from this Levy process, you can retrieve the law of age of the node depths. And also, if you add mass extinctions on top on the initial process, you can deduce the expression of f thanks to the initial expression of f after mass extinction. Now, so let me show you three papers that use this machinery to infer the diversification process from the mere knowledge of the phylogeny. So the first of these papers is a paper by my colleagues Hélène Morlon and Todd Parsons, and also Joshua Plotkin, where B and D only depend on time, parameterized, I think, by exponentials. And what they did here is that they took the phylogeny of cetaceans, and from this phylogeny, they inferred how B and D had depended on time in the past. And then on the right, what they did is that they simulated the birth-death process with the B and D that they had inferred, and they obtained this shape of boom and bust from the number of species through time in the past. And you see that they actually recover quite well what was known from the fossil record, an initially increasing number of species up to 200 to 250 species, and then contracting down to the 86 species of cetaceans that are known today. The second example is a sole author paper by Tanya Stadler. She used the phylogeny of mammals that I showed you earlier. Actually, she broke this phylogeny into several subtrees, and she inferred the way B and D depended on time in a piecewise fashion for each of these subtrees. In blue, you see what the result of the inference for the whole phylogeny. The most important and best statistically supported shifts in diversification. So here you see on the y-axis the diversification rate, which is just B minus D, the growth rate of the phylogeny, the growth rate of the number of species. And you can see that there has been a great peak in all these subclades, in all these taxa, a peak approximately 30 million years ago, coinciding with the cooling of the Earth. And the other best statistically supported shift is 3.3 million years ago. You see a sharp decrease in diversification, coinciding with high tectonic activity. In a third work where I was involved with Tanya Stadler and Helen Alexander, instead of having B and D depend on time, we assumed that B was constant and D depending on age, meaning, as I said earlier, that the lifetime of a species could be anything, and we parameterized it in terms of a gamma random variable with shape parameter k. And what we did is that we used we first checked whether the coalescent point process framework was able to accurately estimate by maximum likelihood. This is what MLE stands for. So maximum likelihood estimates. If we could accurately estimate B, K, and S, and this worked. So we applied the method to the bird phylogeny that I showed you previously, with close to 10,000 species. And the result is that the exponential model, which corresponds to k equals 1, k equals 1 means that the lifetime is exponential, and so that D does not depend on age. The exponential model is rejected with a large statistical support. And so the extinction rate means that the extinction rate depends on age. And because the shape parameter was actually estimated to be much larger than 1, it means that the extinction rate increases with age, suggesting that species do age. In passing, we estimated the average lifetime of species to be approximately 15 million years, and that each species gave on average birth to another species every 10 to the 5 years, 0.1 million years. All right, so these lineage-based models of macroevolution are very popular. There are all sorts of such models. So I showed you the time-dependent models. I showed you an age-dependent example. There is also well-known work on diversity-dependent diversification, where the rates depend on N. And there is since 2007, and this paper by Madison et al., there has been a craze about trade-dependent diversification and the methods that are associated with these models: BC for a binary state, quasi for a quantitative state, high C for a hidden state. And actually, these methods have been so popular in the last decade that you could read up to 4 survey articles on this same topic. So I show here on the right the titles of these 4 popular survey articles. But actually, the problem is that, of course, species are not particles. Okay, and so if you want to get some information about the diversification process, a little more than just the speciation rate or extinction rate, which does not have, like, let's say, a biological meaning, at least in terms of what you can measure on the field. These models do not inform us about these. And so, and in addition, it is known that reproductive isolation is progressive, it takes time, and so speciation is not instantaneous as in these models. You see on the cartoons here 2 examples. So the first example is known as allopatric speciation, where 2 populations initially coming from the same species are separated geographically, and reproductive isolation occurs as a byproduct of local adaptation. The second cartoon is actually a toy example explaining why this can happen. It's called Betson-Dobzhansky-Muller incompatibilities, BDM. It's supposed to model what happens at the genetic level. And so if you can see if you can read the cartoon on the right, you see that you start with a one monomorphic population where at locus 1 everybody carries small a allele, so small a because it's a diploid species. At locus 2 everybody carries the small b allele, and then this population splits into 2. And as time evolves, the first population sees a mutation from small a to capital A in some individual, and maybe because of local adaptation, this big A allele will go to fixation until everybody is homozygous at this locus, big A. And the same occurs at locus 2 in the second population. And so at this point, you have one population which carries big A, small b, the other population which carries small a, big B. And so the hybrids coming from individuals, the hybrids of individuals coming from these 2 populations will be the double heterozygotes, capital A, capital B, small b. If this double heterozygote is unviable, then you have produced two populations that are reproductively isolated from each other. Of course, this takes time, but you see that it kind of solves what is called the species paradox, the fact that at no point in time there has been a first individual of a new species. So in what's next, I want to show how we can try at least to open the species box and propose some, let's say, simple and generic models for the progressive emergence of reproductive isolation. So the idea is that now in these models, species are no longer the elementary particles in the system. The elementary particles can be individuals, but I will more often talk about populations. And these populations can replicate when founders from the seed population occupy and colonize a new habitat and found a new population. They can die, of course, when there is local extinction of a population. And we assume that it can mutate. So now mutations are not like a single substitutions as we saw last week, but more think of mutations as a major genetic change or a new stage in the speciation process that gets you closer or gets the population closer from a new species. And now what I will assume is that speciation is just a consequence of these phenotypic or genetic changes and differentiation between populations. And now compared to the lineage-based models where we very simply saw that we very simply had new species occurring and very easy to define the species phylogeny, and we had a very efficient way of simulating and inferring from the phylogeny. Now we need to have a natural way of partitioning the particles, the populations, into species according to their phenotypes or their genetic differences. A unique way of defining the species phylogeny consistently with the genealogy of particles, meaning that if particles or populations are closely related, then they should be in the same species, and if they are distantly related, they should be in different species. A fast algorithm simulating the partition and the phylogeny, and a statistical method for the inference of the microscopic parameters of the process. In the lineage-based models, we had A, B, C, D for free, and now we have to check that we can get these items in our model. So let me present you three, let's say, embryonic works in this direction, which are attempts of modeling speciation from, let's say, first principle. So the first example is called the protracted speciation, has been proposed by James Rosendale and his co-authors. And so his idea was that speciation was not instantaneous; it takes time, and that species can be seen as an ensemble of populations, as I just said, and that each population gradually diverges from mother species. And so the idea is very simple: is that these populations, as I said, go through mutations, they accumulate mutations, and speciation is complete when the population has accumulated K mutations. Okay, so K is the threshold for the new species to arise. The newborn particles are called incipient. The first stage in which they are born is called incipient, means that they belong to the same species as the mother population. And after having accumulated K mutations, they arrive in a stage which is called good, meaning that they now start a new species. So that's for A. It induces a natural partition of particles into species. And now for the phylogeny, what we did, what Rampal Etienne, our co-author, did in the work that I will now present, we will assume that each species is actually represented by one single particle. And so the phylogeny is defined, that's for B, as the tree in the genealogy of particles spanned by these representative particles. Okay, so now we have solved A and B, and now we'll see how we can do C and D. So how can we choose these representative particles? Here you see an example of a tree of particles where the dashed lines, so the time flows upwards. Dashed lines are incipient stage, and the solid lines mean that these populations are in the good stage. They are a new species. So here K equals 1. And you see that at time capital T, there are 4 populations alive. Population 1 is in the good stage. It's a brand new species. Population 4 is in the incipient stage, but since it's the only representative of species B, species B is represented by this population, population 4. And you see that species A is represented or has 2 descending incipient species at time capital T. And what we chose is to assume that the representative population of species A is the leftmost particle in the natural tree orientation. And so species A is represented by population 2. If we make this assumption, and if we assume that the population birth rate does not depend on speciation stage, then we could show in the paper that is mentioned on top of the slide that the tree spanned by the representative particles, one per species, the leftmost in the tree, is a coalescent point process with explicit node depth distribution. And so we are back to what we know, how to simulate very easily and how to infer, because the phylogeny is again a coalescent point process, but now parameterized by the microscopic parameters or the microscopic ingredients of this protracted speciation model. So what we've done then is to try to apply this method and to apply this model to real data. So we first tested the model on simulations, and we actually realized that we could not infer the individual parameters of the model, but that we could infer a composite parameter, which is the duration of speciation. So what's the duration of speciation in this framework? It's the waiting time until an incipient species has a good population in its descendants. So for example, if I go back to the previous slide, you see that the time at which the population B arises is the time it took for speciation to occur. And so we applied this by breaking down the bird phylogeny into 46 bird clades. And you see on the left the results of the analysis, where we infer this duration of speciation under the protracted speciation model for each of these 46 bird clades. And you see this very nice range of estimates going from 10,000 years to 10 million years. And these estimates are actually in agreement with the estimates that I showed you with the age-dependent model earlier. So so let me now introduce model 2. So model 2 starts from the realization that we don't have any knowledge of who is the mother species, which is actually a problem which is formally the same as guessing the ancestral state when you have a sample of DNA sequences. And so because we don't have the knowledge of the mother species, we cannot really partition particles with reference to this mother species, and we can only sort particles in different species according to their differences. Okay, and so the two natural rules that you would like to apply in this framework are the following. For example, you can fix some threshold Q, and rule 1 is to consist in putting in the same species all particles that are separated by less than 2 mutations. And rule 2 is basically the converse, saying that particles separated by more than 2 mutations are in different species. Of course, what you would like is to have a partition in 2 species which satisfies the 2 rules simultaneously. But if you look at the example on the right and you take Q equals 1, you will see, for example, that if you apply rule 1, particle 1 and 3 will be in the same species because they are separated by less than 1 mutation. 3 and 7 will be in the same species because they are separated by no mutation. 7 and 9 will be in the same species because they are separated by 1 mutation. And so 1 and 9 will be in the same species.
So these X is labeled by alpha, beta, gamma, delta are the mutations?
They are just mutations that occur, let's say, at different places on the genome. But yeah, maybe forget, it's just to say that there are different mutations occurring at different loci. And so you see that if you apply rule 1 and 9 will be in the same species, but this contradicts rule 2. And so there's no way actually of reconciling the 2 rules. And actually, even if you only take one of these rules, there are multiple partitions that satisfy either rule. For example, rule 1, the partition where you put all particles in the same species satisfies rule 1, and the partition where you put every single particle in a different species satisfies rule 2. Okay, so it's not clear how you can partition into species. That's problem A, and even less clear if you want to define the phylogeny from this partition. Now, a nice situation happens when each subset of tips in your partition is monophyletic. What does it mean? Monophyletic means that it forms a subtree. Okay, so it includes all the descendants from their most recent common ancestor in the genealogy. And it's not easy, it's not difficult to see that each species form monophyletic subsets. Then there is a unique phylogeny that can be consistent with the genealogy, and that's obtained by collapsing each subset of your partition into one single tip in the phylogeny. And the good news is that we can define naturally some monophyletic partitions that satisfy the rules that I just stated. So that's a result obtained with Marc Manceau, a former PhD of mine, co-supervised by Hélène Morlon. And the result says that there is a unique species partition that is the finest monophyletic partition satisfying rule 1. And there's also a unique species partition that is the coarsest monophyletic partition satisfying rule 2.
Could you explain again what monophyletic means?
So monophyletic means it's a property of a subset of tips which are related by a tree. And monophyletic means that this subset forms a subtree in the genealogy. Otherwise said, it says that this subset contains all the descendants from the most recent common ancestor of the subset. For example, 6 and 7 is monophyletic. 4, 5, 6, and 7 is monophyletic. But 6, 7, and 8 is not monophyletic. That's clear?
Yeah.
So we use that first rule to apply this model to real data. So forgive me about this dense slide. We started with an individual-based birth-death process with rates B and D for the particles, Poisson mutations at rate theta, and then we defined the species and the phylogeny from the definition that I just gave, the finest monophyletic partition, such that 2 clonal tips belong to the same species. That's Q equals 1, rule 1. That's for A and B, so we have a nice way of partitioning into species and defining the phylogeny from this monophyletic partition. And now for C, we showed that actually, if instead of bookkeeping all the mutations in the genealogy, you only keep the information of state 1 or 0 of the lineage, which actually is saying that a lineage is in state 1 if the type that it's carrying at the time you're looking at it is not represented at time capital T, and state 0 if the type it carries will be represented in the future at time capital T. Then with this 3 type if you enlarge the information of the genealogy with these states, then you can simulate very rapidly the phylogeny by collapsing into one tip all the descendants of each node where the 2 descending subtrees are in state 0. And then for D, we had what is known as a peeling algorithm, which is a way of, let's say, dividing and conquering let's say the exploration of the tree and computing the likelihood of a given phylogeny under the model. And what we showed is that when we apply this method to simulations, we get very precise estimates, as you can see on the well-behaved contour likelihood contour plot on the right, very precise estimates of 2 parameters, theta, the mutation rate, and B minus D, the growth rate of the genealogy. And at the end of the lecture, I will show you a nice application of this model. So the third model is called the split and drift evolving graph. And if you recall what we just did in model 2, you could actually imagine that what we did is actually draw an edge between particles that are separated by less than Q differences. And the idea is that basically the species are the connected components of the graph that you define this way. The fact of knowing whether an edge exists or not depends on the whole genealogy and the mutations that are on it. But our idea was to now explicitly model these edges. And in the framework of systematics and the biological definition of species, then you declare that an edge is present between particles as soon as they are able to interbreed. And our goal here was to have a simple model where interbreeding, these edges, evolve by replication when the when the particles split, and spontaneous divergence that we call drift when the edges, after some random time, spontaneously disappear. Note that this interbreeding relationship is not transitive, as you can see from this example here, a well-known example of ring species, starting with so that's species from the genus Ensatina of salamanders. It starts with the one ancestral species in the northwest of the USA, and you see that it diversified into different morphs on the two sides of the San Joaquin Valley. And here, two adjacent populations can interbreed, but when the populations meet on the southern side of the San Joaquin Valley, the two populations here cannot interbreed. So you see that this interbreeding graph is not transitive. It's cyclic, minus one edge at the southern end of the valley. And so what we would like to do is to define species as the connected component of the interbreeding graph. So the model here is as follows. It's again a model where there are births, there are deaths, and there are mutations. But mutations are actually a little bit simpler. They just involve edges that disappear after some random time. So here we assume that the number of populations, the number of particles is fixed, equal to n. And the birth and deaths occur according to a Moran model, which is actually the counterpart to the Wright-Fisher model in continuous time. And this goes as follows. I treat 1/2 per oriented pair AB of particles or populations. Population B goes extinct, and simultaneously A gives birth to a new population called A prime. And now how does the graph evolve at that time? Since A prime is the daughter of A, it's assumed to be clonal to A, and so it's assumed to be able to interbreed with all the neighbors of A. So you copy all these edges, and you add a copy between A and A prime, the mother and the daughter, because they're clonal to each other, they're assumed to be able to interbreed. And a divergence is just assuming that each edge disappears after an exponential time with parameter R. So in this paper that you can see the title on top of the slide, we did not really study the phylogeny, but we studied the stationary state of this process, which is an irreducible Markov chain on a finite space of graphs with n vertices. And so it's a very simple two-parameter model where n is the total capacity of the meta-population, and R is the rate of evolution of reproductive isolation. So as far as a simulation is concerned, we can use the fact that the genealogy of particles is given by the Kingman coalescent that you can remember from last week's lecture. And the presence of an edge between I and J depends on a Poisson process with rate R on the pair IJ. And so if there's an atom of this Poisson point process that has occurred before the time to most recent common ancestor of this pair of particles, then the edge has disappeared. And so from these two independent, very simple objects, we can define and fast simulate the graph at stationarity. Now we can also derive some mathematical results on this graph.
Could you go back to the last one? I just want to make sure I understood. So you take two independent things, one which is a tree, something like a Kingman coalescence, and the other which is a labeling on the pairs IJ. And then how do you put this together?
So how we put this together is that so if you have an edge between I and J, it was born at the time of birth. Okay, it was born at a coalescence event, if you look backwards in time, between I and J. Okay, and so this edge is still alive if the Poisson point process labeled IJ has no atom since the coalescence time.
Okay.
Okay.
So
Every edge appears at rate R.
the 2-3 connection remains, but the 1-2 doesn't, I guess. Right.
Exactly, the 1-2 doesn't, and the 1-3 does.
Mm-hmm.
Okay, because the red cross here is upstream of the coalescence time between 1 and 3.
Okay.
Okay. And so some simple mathematical results that you can get using this picture, it goes as follows. If you fix k nodes in the graph, and you're interested in knowing whether all these k nodes, if all the edges are still present, then by a standard argument of competing clocks, the probability that these k nodes form a clique or a complete subgraph is the probability that between two coalescence events, none of the edges has disappeared. Okay, and so it's simple to calculate this as 1/1 plus r to the k minus 1. And in particular, if you take k equals 2, then p2 of n and r is the probability of edge presence between these two nodes. And so you can get a first sense of how the degree of a fixed node depends on the parameters. The expectation of D is n minus 1, the number of other nodes, multiplied by P2, 1/1 plus R. And so this behaves as n over R as n and R go to infinity. So what we did next was to check whether D of n and R rescaled by this parameter converged. And here's what we got. So if D stands for the degree of a fixed node and CC for the number of connected components in the graph, what we obtain is that assuming that n as n goes to infinity, R goes to infinity and R over n goes to 0, indeed, D rescaled by n over R converges in distribution to a size-biased exponential variable with parameter 2. And we also obtained convergence. We also obtained actually an explicit formula for the distribution of the law of D for fixed n and R. And we obtained the tightness, not the convergence, of the number of connected components divided by R. And so you can see a simulation on the right where n is large, R is large but much smaller than n. And you see that you have approximately n over R connected components where the degree of each node is of the order, R connected components, and each node is connected to n over R other nodes. And so you see that, for example, this very simple model gives a very clear prediction on the number of species, and it says that basically this number of species should not depend on the total number of individuals or particles in the metapopulation, but only on the rate of reproductive isolation. And so I think that here with this model, we have a quite promising model, which is highly tractable, neutral in the sense that it does not involve selection. For the evolution of reproductive isolation, there are lots of open questions remaining. For example, the convergence in distribution of this number of connected components divided by R, the distribution of all the sizes of connected components, maybe also the convergence in the Graphon sense, in the dense regime when R is constant and the degree of a fixed node is of the order of n. And as you see, we did not at all tackle the question of how to define, simulate, or characterize mathematically the phylogeny. But there is a very natural way of defining the phylogeny because connected components have a genealogy. If you think of extinction, a connected component dies when the last individual of the connected component dies, and it gives birth when two connected components are, for example, linked by a single edge, and this edge disappears. At this point, you have birth of two connected components. And of course, we're far from making inferences, but this model has very general predictions, and not only in terms of the phylogeny and the number of species, but also in terms of abundances of species. And this is something that is currently in progress with François Bienvenu, who is a PhD student who defended a year ago. So you see the various simulations here on the bottom. So for those interested, I would be glad to take or to share your ideas, our ideas.
But just to make sure I understand, so you take this model that you were describing where you have a coalescence and then you have these point processes, you create your random graph in this way. You look for this sort of stationary measure for this process in time. And this is what you're studying then, the large N limit, or the large number of particle limit. And you're finding these sort of cool features.
Yeah, exactly. Yeah.
Okay.
Exactly. So what I showed here on this picture is really the stationary state of the graph. But these connected components have some evolutionary history. They have a file. They are really like related by a phylogeny, a genealogy, and this is something I would like also to study. So in the abstract, I had mentioned two applications. One was this question of how does the interbreeding graph look like at stationarity. And the second one, for the 5 minutes remaining, is, let's say, a deeper dive into this question of beta and the imbalance of tree shapes. And actually, this question goes back to works and questions by the famous probabilist David Aldous, who came up with a model for a random binary tree T sub n with n exchangeable tips labeled by 1n. Okay, so what's the model that he proposed? So he assumed that we are given distributions Qn on 1n minus 1. And we are given n balls which are colored and actually labeled by 1n. So you start from the bottom of the picture. You have these 6 colored balls. You draw some variable kn, k6, according to the distribution Qn. You find 4, and then you randomly sample uniformly 4 of these balls. You put them into one box, and the 2 others into another box. Okay, and you do that again and again, independently, until every colored dot, every ball, is in a single, is in a different box. When you do that, once you have done that, you can actually copy that and apply the same procedure to a tree, right? So meaning that when you divide your colored balls into a box with 4 and a box with 2, you can say that you will share the labels, the corresponding labels, to the left part in the left subtree and the 2 others in the right subtree. Left and right having no importance here, right? And so you do that again and again, and you produce a binary tree where all the labels play the same role, they're exchangeable, and with n such labels. Okay? Is it clear? So you do that using Qn first, and then Q4, so Q6 first, then Q4 and Q2, then Q3, etc. Okay? And a nice feature of this model is that when you take Qn uniform, it yields exactly the same tree shape as a Yule tree stopped at a fixed time, and also as the Kingman coalescent. Now, if you remember, I talked about sampling consistency last week. So recall that Tn is a random binary tree with n exchangeable tips labeled by 1n. And if you call T prime n the tree obtained by removing one tip from Tn plus 1, say the tip labeled n plus 1, but they play the same role, then the model is said sampling consistent if Tn and T prime n have the same distribution. Of course, this property is quite desirable in biology. You don't want to have your sample depend on the order in which you have sampled your individuals, and that not depend on if you have like sampled let's say n plus 1 and then removed 1. You don't want this sample to be different from just taking n from the beginning. And of course, the archetypal example is the Kingman coalescent. So a way of constructing a sampling consistent model is as follows. So assume that we're given some measure or some law mu on the unit interval. Okay, and so here on the right you see the unit interval. So what you do is that first you uniformly distribute your color dots in the unit interval. Okay, so that's the bottom of the picture. Okay, so you uniformly distribute them independently in the unit interval. And then the first thing that you do is that you fragment the unit interval according to the measure mu. That's the cut point drawn in red here on the picture. And then you consider separately each of the two intervals, and that makes a sharing between the balls or the dots that are on the left and the dots that are on the right. Okay, and you do that again and again until all the dots are in different subintervals of the unit interval, and that produces a tree. And why is that sampling consistent? Because of course, if you, for example, look at the subtree here, and whether you can generate the subtree like starting with 5 colors or six colors, and then take away 1 color, you get the same tree, right? Actually, you can even couple all these trees in the same state, in the same space, same probability space, by throwing all the integers from the beginning in the unit interval. Actually, the converse was proved by Haas, Miermont, Pittman, and Winkel in 2008 using fragmentation processes, and I did the same but without using fragmentation processes recently, and they showed that this tree model is sampling consistent if and only if we are in this situation where, in which case Qn of I can be written as on the red, with the red expression here on the bottom of the picture. Actually, you can. There's a more general model where you can add what is called erosion, meaning that sometimes you will put one of these dots at random in an isolated box. And the nice thing is that when Aldous applied this model to real data, so he parameterized the measure mu by the beta distribution, so a symmetric density x to the beta, 1 minus x to the beta. And so that imbalance decreases with beta. He showed, and Bloom and François a few years later, showed that the beta that you estimate from empirical trees is not at all close to 0, as in the Yule tree or in the Kingman coalescent, but it's centered on -1. And so the question that still remains up until today that he asked 20 years ago was if there are mathematically simple or biologically plausible stochastic models for phylogenetic trees whose realizations mimic this property of having this imbalance of beta close to -1 rather than close to 0. And so I want to give here, and then it will be over. I want to give here a few partial answers to that question. So first, if you remember one of the first slides, I showed you that when you look at birth-death processes, the reconstructed tree always has the same tree shape as beta equals 0, whenever B only depends on T and N, and D only depends on T, N, and A. So in this whole class of birth-death models, you will never generate reconstructed trees which have the imbalance observed in empirical trees. Now, if you look at the models that are introduced afterwards, model 1, prototic speciation, also produces coalescent point processes, and so they have the same tree shape as beta equals 0 again. Now, if you look at model 2, speciation with genetic differentiation, you can see on the right panel, top right, that the simulations that we made with our model, with the parameters that we had inferred from real data, are similar to the real values of the statistics that are observed in empirical trees. So it means that one possibility is that this neutral model where you cluster individuals which are of the same type, provided they are in the same subtree, which mimics the way systematicians partition populations into species, might be responsible for this empirical imbalance. Another simple explanation, which has been proposed in 2015, is that speciation rate depends on age. So in that paper here that I cited here in the bottom, B depends on age, like a power function, a to the phi minus 1. And what they did is that they estimated phi on more than 9,000 empirical species trees from Treebase, a famous database. They saw that phi was between 0 and 1, indicating that speciation rate decreases with age, so that species do age. And the distribution of beta that is generated by these phi estimates, as you can see on the bottom panel, fits well the distribution that I showed you here in Bloom and François' paper. So that might be an alternative explanation for this imbalance observed empirically. And that's all. I want to thank my co-authors on all these papers that I rapidly presented, especially so Marc Manceau and François Bienvenu, who are PhD students, very talented PhD students in the group, and their co-supervisors, Hélène Morlon and Florence Desbarres. And I give you the few references that you can browse on the recording. And I thank you very much for your patience.
Well, thank you very much, Amaury. Everyone should be able to unmute themselves, so if you want to join with me in a round of applause. And we can take a bit of time, but maybe if people need to leave, they can go as well. If people have questions, and I know that there were some in the chat, I'll just mention the most recent, which is from Francois Bonhomme. How do you deal with the secondary gene flow after incomplete RI? Or how do you deal with incomplete RI because you have a very simple threshold to define RI? Maybe you can remind us what RI means as well.
So RI is a reproductive isolation. And yeah, so actually I do not allow in the models, I do not allow how do you call that, reversing isolation. So you can only increase reproductive isolation by spontaneous divergence between particles. So I never like model a secondary contact or let's say reversal to no reproductive isolation.
Yeah, there's another question: is selection an alternative explanation for beta equaling -1?
Yeah, so the problem is that it's hard to really like picture how selection occurs at the species level, right? So of course you might assume that a trait like I, a heritable trait, acts at the species selection level, meaning that, for example, species which have like a smaller body mass speciate more than others, then of course you will produce a more imbalanced substrate. Yeah, sure. So species selection can be an explanation for imbalance.
Okay. Well, so maybe now is a good time to end. Again, we thank Amaury for his excellent lecture, and we'll post the recording and the slides on the website, a similar link to the one that we gave before, so the IICD website. And we'll also send around a reminder for the third talk, which will occur same time, same place next week. And then we'll have a 1 week gap, and then we'll start up with Alison Etheridge's talks. Okay, well, good to see everyone.
Thank you very much. Have a nice weekend. See you next week.
Yep.
Bye.